So in textbook, synapses are usually modeled as like 101, meaning that like one neuron is connecting another neuron in just one synapses. However, we know that this view is very simplified. One of the cell types that doesn't fall into this simple picture is astrocytes, that they are usually like connecting a bunch of other neurons and have very weird morphologies. To understand the rule and the function, we need to model their network. Today, Marco is presenting a mathematical framework for neuron-glia network. Marco Peña-Garcia is a post-bachelor researcher associate from San Marcos University, working with Dr. Walter Cabrera. And today he's going to talk about mathematical representation of neuron-glia networks. Thank you again, Marco, for joining us today. And we are ready to hear your interesting talk.
Okay, I will share my screen.
Great.
Thanks for accept my abstract. I will expose a mathematical framework as is in the title, mathematical representation of neuron-glia networks. Well, the brain has more than neurons. The brain has astrocytes, and we know that astrocytes are also implied in cognitive computations. Well, as a cartoon of this complex network, I have some diagrams. Well, the basis of neuron-glia networks is that tripartite synapses. That implies that a neuron interact with another neuron, but also with astrocytes. Neurons synthesize neurotransmitter, release neurotransmitter, and are received by the other neuron. But also astrocytes have neuroreceptors, and astrocytes also have a response to this neurotransmitter, increasing the calcium levels, the cytoplasmic calcium levels, and there are some waves of calcium that travel along one astrocyte to another astrocyte, and that's the astrocyte network. Astrocyte has have networks also. These networks not have synapses in a classical sense, but have gap junctions, and the gap junctions are quite similar to electrical synapses because it allows some signals to travel along two cells, IP3 calcium, but also astrocytes release neurotransmitters, release ATP, glutamate, and can activate neurons also. So it's a complex network with interaction from one side to the another side and vice versa. So if you want more information about astrocyte networks, neuron-glia network, you can read the book of Dipita, more information about computational glia science. Now, mathematically, I will be not too formal because I know this is not a Congress of Mathematicians. I will be more informal. So we have astrocytes and neurons, but to model the network as a whole, we need a term for both. And the term is, a suggestion is general neuron, I think, because astrocyte also synthesize neurotransmitter, is affected and receive neurotransmitters, so it's like behaving like a neuron. So it's a general neuron. Well, and we need a set for this general neuron. We call the set X, the GN set. And we have that this set is the union of the set of neurons and the set of astrocytes. And now we have to formalize the interaction, the complex interaction between astrocyte neurons or neurons and neurons or astrocyte and astrocytes. And we need a general framework for that. So we can see synapses as the graph of a binary relation. What's a binary relation? A binary relation is an ordered triple that has a departure set. Is like a function. The function has a domain and range. It's something very similar in relations. Relation has a departure set and then destiny set and a graph that model how they are interacting. And this graph is a subset of the Cartesian product between A and B. In this case, we have only one set the set of general neurons. So A equal to B, and A is included, it's a subset of X. So this A is the set of interacting general neurons. It's like all neurons that are interacting like this. In this case, we have two neurons and astrocyte. This set has the seed could be one neuron, another neuron, and the astrocytes. And in simple words, we have this graph. This graph is simply a set of ordered pairs like this. It's an ordered pair with one general neuron and another general neuron. This ordered pair stands for a X neuron X E can set neurotransmitter to neuron XG. It's as simple as that. And the set has all the interactions in these synapses that can be a tripartite synapses, a simple synapses, or a gap junction between astrocytes. Well, in this way, we define all of these synapses. As an example, a gap junction. A gap junction is a synapses and that allow the flows from one neuron to another neuron and vice versa. So this graph has both direction represented by these ordered pairs. And that's a bit formal, but we can think in synapses as these sets of ordered pairs, meaning the interactions. But we have more than one synapses between two neurons. As we know, a neuron can be hundred of synapses between this neuron to another neuron. Some in one way and some others in another way. So we need a framework that cover this complexity. And we have the term in mathematics that index family. What's an index family? Oh, another time, and triple. Another triple that has A, that A is the set of all synapses, and I is the set of indexes. So we have that indexes could be 1, 2, 3 until the end, and F is a surjective mapping, a surjective function from I to A. So graphically, more easy to understand, we have this pair that is one synapse. Remember, in this case, one synapse is a simple synapses, one-to-one direct synapses, classical synapses. And we have these indexes. So we have, in this sense, in this direction, from xi to xg, we have three synapses. So we have three indexes also in the indexes set. So we have xe repeated 3 times, but these are different synapses. And another direction, we have the 4 synapses in this case, another direction synapses. But we have some conditions. First, the condition that we have at least one tripartite synapses, the minimal condition, because this is a neuron-glia network, so we need a tripartite synapses. Another condition is that we can define a sequence from one neuron to any other neurons that belong to a graph in the set of synapses, or the inverted graph. That is that I can I can do this path in this direction if these synapses are in this direction, but I can do that another way, because this inverted graph is like inverting all direction of the network, including the tripartite synapses that is much more complex than simple synapses. But I can invert all these directions with the term of inverted graphs. So, but we can have one neuron that I can reach. There are no synapses, so this is one condition to wholeness. We have one network, not unconnected graph. Then we can see also this complexity as not as an indexed family that is a bit complex. We can simplify it a bit using multi-set of synapses. A multiset of synapses say, well, I have 3 synapses in this direction and 1 synapses in another direction, and we have a counting function that says that this direction, this synapse has 3 times appear 3 times in the set. So it and another direction 1 time. This is like a counting function. This concept is quite similar to weighted graph, but it's not weighted graph. It's something more general than weighted graph, because in multisets, each element literally appeared more than 1 time. It's not like a function, only a function. No, it's that the multiset has this element repeated way, many times, n times. So we have this to simplify a bit. It's like a simplification, because we have this synapse repeated 3 times. It's not like we have 3 synapses, but the same synapses actually. In an indexing function, indexing family, we have these synapses appearing 3 times, but there are different synapses in that mathematical sense. In this case, we have the same synapses appearing 3 times. It's like something like that. If you want to see more about multiset, you can read to Girish. It's a Girish 2009 and all his material. Well, we want to simplify a bit more from multiset to multidigraph. Probably you know about digraphs. Multidigraph is just repeating these simple connections many times. If we have multisets like this, we are complicating a bit more. Now we have a gap junction here appearing 3 times, and a simple synapses appearing 1 time in the multiset. We want to transform this multiset in a multidigraph. We need to unnest the multiset and only put in some directions that appear 4 times and another direction appear 1 time. And this is a multidigraph. It has some advantage and disadvantage. The advantage is that multidigraph as graph has many properties and are very useful in neuroscience, use graph and digraph for network science. And the disadvantage is that we conjecture that it doesn't perceive isomorphisms. In graph and in general, in category theory, we have a term called isomorphism. Isomorphism, informally, is a structure preserving mapping between 2 structures. As an example, we have these 2 graphs and are isomorphic. It looks so different, but are the same thing. The same graph in this with different levels. And the same thing can be defined for multidigraph and upsilon set also. It's just formalizing and generalizing the concept of isomorphism in graphs to multidigraph and to multisets. But we conjecture that this transformation does not preserve isomorphism in general. We are working now to know in which case preserve isomorphism, because transforming a multigraph have lots of advantages, and we are working on know in which case it could be work. So I'm not sure if I have more time for the extra results.
Can you wrap it up soon, Marco? We have 2 minutes overall.
Okay, we have 2 minutes. Well, we have
including questions. Sorry, including question 2 minutes.
Oh, okay. Yeah, that's all in this case. Well, and acknowledge to my collaborator Nelson Castro from Brazil, IMPAM. Thanks.
Thank you, Marco. That was great. Yeah, feel free to ask question in Q&A. Marco, I think I missed the part that you were talking about astrocytes. What is a specific thing about astrocyte in this modeling?
Yeah, the astrocyte play a role in the network. It's like these astrocytes are connected to single synapses. That's the tripartite synapses. So we want to model this network with these multiple connections. That was the goal of the study.
But you can use it for any neural network, right? For any sort of...
in general, you can use this to model with physics or to connectomics, for example. Now we have lots of information about connection between synapses, but we can also reconstruct astrocytes and look their connections and whether the astrocytes are connected with a single synapses is not too difficult, but we need a framework to model all the network and not only neurons. If you have to model only neurons, you can use graph theories or digraph theory. But if you want to include astrocytes, we need other thing that is not a graph, definitely. Could be an index family, could be a multisets. And we can simplify to a digraph, but we are losing some properties. That's...
Thank you so much. Thank you so much. That was great, Taika, and I am looking forward to your future work. Perfect. Thank you so much. And let me go to the lab.